Good morning. I'm Brad Hyman. I'm director of the Massachusetts Alzheimer's Disease Research Center, a neurologist and a neuroscientist up here in Boston. I am glad to have the opportunity to participate today, and I'd like to talk about determining the fate of Alzheimer's patients, thinking about heterogeneity and what that means in terms of personalized medicine. First, though, I'll apologize that I'm not there in person. You can see the reason by the cartoon. A series of disclosures. I've had the good fortune to work with several companies over the years. I'd also like to especially acknowledge the lab members who contributed to the work that I'm going to be presenting, and I'll highlight work that Simone Dujardin and Alberto Serrano Pozo especially contributed to over the past few years. Acknowledge with appreciation the funding sources, especially, of course, the NIH, and highlight also the work from our collaborators, Judith Steen's lab at Children's Hospital in Boston, and then also our collaborators in industry, Merck and Lilly. The question I'd like to just briefly address here, and as a way of kicking off the topic, is why do some patients have rapid, severe disease and others a slow progression or even a stable, mild course? I have patients in the clinic who, when I see originally, will have an identical presentation, a little bit of memory problems, some concerns about what's been going on for the last 6 months or a year, slowly progressive illness, maybe a family history, and yet one individual might go on and over the course of the next 2 years develop a severe dementia which precludes them from even finding their way around their own house, and the other individual might a decade later still be coming in to see me and be able to converse about day-to-day events. What are the contributors to this variation between one individual to the next? I'm going to highlight 4 potential contributors. The possibility that the tau that's present in neurofibrillary tangles in the brain of one Alzheimer's patient is not the same as the tau in another brain, and that these tau strains connote or correlate with differences in the clinically important variables such as age of onset and rate of progression. I'll also highlight genetics, especially the apolipoprotein E genotype. This is, of course, the predominant genotype and risk factor for Alzheimer's disease. Finally, a broader genetics perspective, thinking that there are other genes that may well be important in determining heterogeneity of clinical presentations in Alzheimer's disease. And then I'll end with a brief note to my colleague Teresa Gomez-Isla's work on resilience and neuroinflammation as also playing a major role in how the presence of plaques and tangles and other changes of Alzheimer's disease in the brain manifest in terms of clinical presentations. Now, all of this is in the context of the fact that, sort of obviously, people with Alzheimer's disease are for the most part, fairly elderly. And that by the time you've been on the planet for 60 or 70 or 80 years, it's entirely possible that you have other diseases as well. In fact, in our analysis of a large autopsy series performed in the data set by NAC, which is a national consortium of Alzheimer's centers, we see that of all the autopsies of patients who had dementia, about 71% of them had both Alzheimer's disease and another element seen in neuropathology as being potentially causative of some aspects of cognitive impairment. And only 29%, or about a third of them, had only Alzheimer's disease. And so, of course, it's these only Alzheimer patients that I'm going to be presenting the further data on later, because the heterogeneity that's accounted for by the additional presence of Lewy bodies, of vascular lesions, of TDP-43, of hippocampal sclerosis, and so forth, that in my mind falls into a slightly separate category of heterogeneity. That indeed, one can always have more than one disease affecting the brain, but if the predominant disease is Alzheimer's disease, even in that instance, there's heterogeneity. This is illustrated by these two spaghetti plots that are shown to the right. If we look at the rate of progression of the clinical dementia rating scale, sum of boxes, this is a commonly used clinical scale of 0 being normal and 18 being severely impaired, you see that the average change in CDR sum of boxes per year for the 8 years or so before death can be calculated, but actually when you plot the original data of the nearly 2,500 patients seen here, it points in all directions. And even if you take out the 71% for the individuals who had Alzheimer's disease plus something else, and you focus only on those Alzheimer's disease that had so-called Alzheimer's alone, you still see that the spaghetti plot has many people who are fast progressors, many people who are slow progressors, and really everything in between. So it's impossible to predict from the very first measure how quickly or slowly someone will progress. And the work we've been doing over the last several years has focused on these Alzheimer's alone cases, proven at autopsy, and then retrospectively asking, what was it about those cases that we might have been able to learn that could contribute to their rate of progression? This illustrates the same sort of graph, the CDR sum of boxes on the y-axis, time on the x-axis, for 32 individuals whom we followed at Mass General Hospital, who were selected because we had followed them for years in clinic, they had come to autopsy, and they were, like the third or so of the individuals from the NAC database, thought to have only Alzheimer's disease as the cause of dementia. And again, this spaghetti plot illustrates that there are some individuals who are fast progressors, some individuals who are slow progressors, other individuals who are somewhere in between. Now, if you think about this amount of heterogeneity is disadvantageous from a clinical point of view for two obvious reasons. The first is that I want to be able to tell a patient what the future holds for them. I want to be able to tell them what it is to expect as the disease progresses. And with this amount of heterogeneity, that becomes an insurmountable challenge. The second is that from a clinical trials perspective, if you're looking for a differentiation between placebo and drug, the amount of noise there is in the placebo group, which is essentially this group, means that the number of individuals you have to follow approaches several thousand. You have to follow them for 18 months to maybe even longer to see a differentiation. And those trials cost $500 million apiece because of the size and length of trial. And that becomes really untenable to do many, many trials and many doses and so forth, or combinations of drugs. And so understanding this heterogeneity, I think, is a really, really important critical problem for the field in order to overcome this practical issue of clinical trial design and the very human issue of trying to serve our patients better. We took a sample of frontal cortex from each of these 32 individuals and performed the bioactivity assay to ask whether or not the tau that was present in these individuals had the same ability to induce aggregation in an endogenous tau environment. And this is an assay that was developed from the Mark Diamond assay that's been widely used. It uses hex cells. And when you can see from the graph on the right that if we map rate of progression, and this is just fastest, slowest, in the individuals from 1 to 32, and the seeding potential, again, lowest to highest, 1 to 32, there's a strong correlation. In fact, we can account for about 15% of the variability in rate of progression based on this single assay. So why would it be that tau in one case appears to act differently in this bioactivity assay, this induction of tau aggregation assay, compared to tau from another individual? We performed mass spec analysis in collaboration with Judith Steen. You'll see on the left for these 32 individuals. There are different patterns of post-translational modifications that are present in each of the 32 individuals, and there seem to be clusters of patterns that accumulate with those individuals who had the fastest rate of progression and the most amount of seeding, and those individuals who had lesser amounts of aggregation or seeding. On the right, you'll see another collaboration with Judith Steen, in which we looked at a much larger group of individuals with Alzheimer's disease, now collected from multiple sites across the country. And again, we saw this remarkable heterogeneity across sites. So, of course, there are multiple phosphosites that are present in essentially every tau molecule of an Alzheimer case. But there are also multiple tau phosphorylation sites, acetylation sites, ubiquitination sites, etc., that are unique to an individual. And it's these unique PTMs which we believe drive the conformational changes that lead to an alteration in the ability to drive aggregation of endogenous tau. In addition to tau, there are other factors that clearly seem to impact the Alzheimer's disease phenotype. Apolipoprotein E comes as E2, E3, and E4 in the general population, with E3 by far predominant, E2 a rare allele, and E4 present in about 15 to 20% of the general population. Alzheimer's disease is of course, overrepresented with E4 individuals, and so two copies of the E4 lead to a roughly 10-fold increased risk for Alzheimer's disease. In addition, a much earlier age of onset. In addition, a faster rate of progression. By contrast, ApoE2 in this study of the large NAC database showed a much later age of progression, or age of onset, and a much slower rate of progression. And when we drill down into that, ApoE2 seems to be especially protective, and perhaps the molecular basis of that is diminished amyloid, diminished tangles, and importantly, diminished neuroinflammation. And I'll be happy to discuss those three possibilities and the role of ApoE in impacting the other aspects of Alzheimer's disease during the question and answers. In addition to ApoE, we reasoned that there may well be other genetic factors important for propagation. We developed an assay using an AAV system in which we could measure propagation of tau from one neuron to the next in animal models. We then examined, in collaboration with our colleagues at Lilly, the amount of propagation that occurred in different mouse models, ranging from the very standard B6, and I'll just as an aside say that the B6, of course, is the mouse strain most commonly used for transgenic animals, to genetically distinct strains We used 5 here in an ongoing study now in collaboration with JAX. We have 19 different strains being used. The story is the same. The amount of propagation seen varies rather dramatically from one strain to the next. And in the JAX strains, we were able to calculate that 40% of the amount of variance in propagation from one animal to the next could be accounted for by the strain background. Now, this mirrors almost exactly the data from identical twins and fraternal twins in humans. That is, the rate of progression, the amount of tau propagation seen in tau PET scans in humans who are either genetically identical or genetically similar, of course, but not identical in fraternal twins. About 40% of that variance is heritable. So it appears as if there are a variety of other genetic factors that will impact propagation and therefore rate of progression that are largely yet to be discovered, but could be found using these human genetic studies and the mouse genetic studies. And I'll close just with a mention of resilience. So in all of our studies of Alzheimer's disease, going really back to 30 years from the very first cases I ever looked at, there was the rare individual who under the microscope had tangles and plaques galore, had plenty of tangles and plaques to meet criteria for Alzheimer's disease. And yet, as best as we could tell from the clinical presentation, had been essentially normal when they passed away. There's a variety of possibilities for this, but this happened, although rare, often enough that we ultimately collected a series of these individuals to study, and it seems as if the tangles and plaques occur, but there's very little microglial response, very little cytokine response, very little astrocytic response. Neuroinflammation in general is tamped down in these individuals, and neuronal and synaptic loss is minimal. And in these rare individuals, whom we refer to, as I say, as resilient individuals, there's the phenotype of tangles and plaques, but an absence of the neural system destruction that underlies the clinical symptoms of Alzheimer's disease. And so this is yet another cause of impairments in Alzheimer's. So in summary, patient heterogeneity confounds clinical care and contributes to difficulties in carrying out and interpreting clinical trials. Our data showed that there are at least 2 and potentially interacting biological correlates of rate of progression. There's this issue of tau strains based on post-translational modifications of tau and potentially mutations and isoforms and so forth that change the conformation of tau and, much like in prion disease, change the clinical phenotype. We also find that ApoE is a genetic risk factor that alters rate of progression, and have data from the mouse studies and from the literature in trials of human twins that there's a strong genetic component to tau propagation as well. So in addition to the comorbid processes like vascular changes and Lewy bodies and so forth, I submit to you the possibility that in order to understand the rate of progression of Alzheimer's disease better for an individual patient, to have precision medicine approaches, we need to have a better understanding of the underlying biology of this heterogeneity that we're just beginning to flesh out. So I want to thank you very much for paying attention. Again, I'm sorry not there in person and obviously not there in full throat. And I'll be happy to try to answer any questions. And I leave my email address here as well in case there's any other issues that you'd like to bring up with me. Thank you so much. So I'm actually here. As you can hear, I'm only partially vocally here, but I'll be happy to try to answer any questions.
Hi, Brad. Great talk. On the ApoE slow progressors versus fast progressors, are you considering also other SNPs? I mean, maybe these might be polygenic ApoE, but in combination with other things. Do we know what other genes might be acting in addition to ApoE on slow versus fast progressors?
Elias, it's a great question, and we don't have a definitive answer. The data I showed you is actually out of the NAC database, so it's sort of the largest one in which there's a uniform series of progressive measures over the course of more than 5 years. There, we have in smaller databases looked to see whether other polymorphisms in the ApoE promoter or whether there was a other SNP that came along for the ride that would drive it, and we don't see anything. But the power of those secondary analyses is probably small unless it was a big effect, and so right now it looks as if the E2 and E3 and E4 are the major drivers, but of course there's the possibility that it's more complex, and that's been one of the themes here. I will say that we can mimic some of those results in transgenic animals, however, and so that at least gives us some more confidence that we're looking at the real biology by looking at the differences between E2, E3, and E4.
Brad, that was a really nice talk. So did I understand correctly, there are a small but significant number of patients who have fairly high levels of plaques and tangles, but in the absence of myeloid cell activation, they don't develop pathology. Did I get that right?
All right, so we'll have to be a little careful about using the term pathology because they've got tangles and plaques, which we would all consider pathology. They have minimal neuronal loss, they have minimal synaptic loss, and they have minimal sort of markers of activation of microglia and astrocytes. And then it's sort of a little bit of chicken and egg there, right? Maybe the microglia are especially activated because synapses are dying back, and in the absence of dying back synapses, the microglia don't get activated. Maybe synapses are dying because microglia are activated. What we know about those individuals is that it's uncommon for them to be E4 positive, fine. And there are some other sort of markers, but they are very unusual individuals, but they're often enough, and we've collected them now from half a dozen collaborating ADRCs across the country, and everybody sees a case like this now and then. It's really remarkable. It gives me faith and hope, at least, that the brain can actually sort of be resilient and recover from the lesions that we have been looking at under the microscope for since 1906.
Indeed, thank you.
So, Brad, very nice talk. So I have a question. So you showed that that E2, E3, E4, at least with respect to the patient, they have different rate of progression. But now, if you take the same E2, E3, E4 patients and then isolate the neurofibrillary tangles from them, do you see any differences at that level? So in other words, if you look at the structure, the conformation, or you look at the activity, for example, of the tangle that you isolate from the different E4 cases, ApoE cases, do you see differences at that level?
Yeah, so you bring up a great point. And the folks who do cryo-EM probably are better addressing this than I, but it's my impression from the cryo-EM data that the final fibrillar structure across ApoE3 versus 4, I don't think there's any E2s that have been done, is identical. But when we isolate the high molecular weight, oligomeric, soluble, I don't know what the right term for this stuff is that we've been, looking at. It's highly post-translationally modified. There's 20 post-translational modifications, and they differ between again, it's just E3s and E4s. We don't have the E2 cases to compare. And the E4 cases on average have a higher bioactivity in this sort of diamond cell aggregation assay than the E3s do. So it does seem as if there's a correlation there between the ApoE genotype and the biophysical characteristics of tau. Whether that correlation is one more thing that happens or is actually the thing that drives it, I think that's still uncertain. And I want to emphasize that the amount of variability that we can sort of correlate with either the tau strain or the ApoE is a relatively small percentage. We're talking 10, 15% of the variability in rates of progression for either of those two variables. But having said that, that's more of the rate of progression than we've been able to identify with any other variables. So the way we interpret that is that the progression in the disease is at least correlates with spread. And that's clinically the case, right? So you start out with memory problems, and then on average, you get language problems, you get visuospatial problems, and so forth. And that correlates where on a tau PET scan or at postmortem, where you see the tau aggregates and they accumulate. And so if we're looking at the diamond assay really as a marker in part of that seeding potential and of the propagation potential for a tau moiety. And so I think it's from that perspective, how likely is it that tau will jump across the synapse and get to the next neural system? It does correlate with rate of progression. At least that's the underlying hypothesis, and has some sort of face validity and plausibility to it. Whether the diamond assay per se tells me something about the structure of the tau that might happen, I think that's a different question. But whether tau, gets into a cell and can corrupt endogenous tau, that's the readout we're using it in context. Yeah, there are two reasons. The first was sort of from a theoretical perspective, it seemed hard to imagine that full fibrils were going to cross synapses. And so from our original work in transgenic mice, where we expressed p-thro and L-tau only in the entorhinal cortex layer 2, by only, I mean, of course, primarily, and then saw the human tau appear in the dentate gyrus, it was clear that was a soluble form that was being secreted and taken up and so forth, because those animals didn't develop the fibrillar form. So we thought that from sort of first principles, it was more likely to be a soluble form that would be transported down axons and potentially released at the synapse. And then secondarily, we've actually now gone back and done a sarcosyl prep and a oligomeric prep from the same set of brains. We've now done eight consecutive brains that came through, and then did both diamond cell assays and also injections into tau transgenic mice or young mice along the way that Virginia Lee has pioneered. And there we see almost identical levels of activity if you normalize for the amount of tau that you've injected. Intriguingly, the sarcosyl tau really has very little activity unless you sonicate it. So it seems as if you have to break it up some, and then it really is, as Virginia has shown, for years now, very, very effective. But sort of just straight out of the brain, so to speak, the high molecular weight is actually more bioactive, and even when you sonicate the sarcosyl material, it's essentially equivalently bioactive. It's interesting. This is work that we don't do to Steen. The PTM out of the sarcocell, and again, it's these same cases where we've isolated sarcocell or the so-called high molecular weight fraction from the same case. And on average, the sarcocell is 40, 50 post-translational modifications, and the high molecular weight has a smaller number on the order of 20, but it's a subset. And so it almost seems as if it's a form frust of the what ends up as a sarcocell fraction. There are a few unique post-translational modifications that occur only, at least with a relatively small n. So I say it's 8 in either the high molecular weight fraction or the sarcocell fraction. But the sort of 20,000 foot view is these high molecular weight fractions a subset of what shows up in the sarc fraction.
I wonder if I can ask a question related to these resilient brains. So because they have deposits, fibrillar deposits, I wonder what they can teach us about the role of these amyloid deposits in disease and in toxicity, and whether you have looked using either the diamond assay or injection or fractionation, whether these deposits are the same as those of patients that do have a cognitive decline and neuronal loss. And if they are the same, then what do you think is the difference? Or maybe they are different.
So that's just, a wonderful question and sort of the, $1 million question in some sense. I'll tell you, what we know about it is that when we just do a high molecular weight in sarc fractionation, it behaves like tau from other cases. We have not done detailed conformational studies. We've, used a standard half dozen antibodies. It behaves the same as tau from, any other Alzheimer cases. It would be intriguing if we had enough material and to think about trying to do a cryo-EM. Maybe there's a subtle difference that we're obviously not picking up with light level, microscopy. But my instinct, and this is a more hypothesis than anything else, is that it's the brain's reaction, it's the neuron's reaction to that material rather than the exact material itself. And that's going to be a tough hypothesis, I think, to demonstrate conclusively. But they really look pretty much the same with the measures that we have at hand. We do now, and they're also multiply ubiquitinated and some fewer number, but still, I think 3 acetylation sites in the high molecular weight. Again, a subset of what you see in the sarc. And so I find the ubiquitin sites especially intriguing. I, sort of was brought up to think that a self-respecting protein that got a ubiquitin on it would go find its way to the proteasome. And these molecules don't. So it's, I couldn't agree with you more. I think we really need to find ways to bring all that information together. And it's, with overlapping structural, really, high quality structural data, I think will be informative. I think you just reinforce all the complexity issues that we've been hearing all day. So, LRP is, of course, a multifaceted receptor, which has many ligands. Ken Kosik's lab and with us, and then more recently with Dudley Strickland, we've demonstrated that LRP actually takes up monomeric and dimeric tau to a better extent than either the high molecular weight or the sarc tau. And so it's clearly a tau receptor, but it has its preferences for what kind of tau it's going to take up. There may well be other tau receptors, and I think there's several groups now, beginning to explore that space. And then you don't even have to think about tau uptake per se. You can think about the consequences of cytokines and other inflammatory mediators on activation of kinases, and then having those kinases act on tau and so forth. So I think as soon as you've got activated microglia and activated astrocytes, for that matter, in the neighborhood, a neuron is going to respond, and it's going to respond in a excuse me, variety of ways. Makes me a little concerned from the talk we heard earlier that COVID-19 is turning on all of my kinases even as we speak. No, so I have to say that I want to thank you all. I was chatting with somebody from my lab a little bit earlier last week, and they were like, "Oh, I'm sorry to be bothering you with these data." And I went, "No, for me, beautiful data are probably the only thing that'll cure me." And so, just having had the honor and pleasure of sitting through the conference today, my fever's gone. My throat is back. I'm, a couple more talks, I'm going to be running marathons. Thank you.